Moving on to a better bee. So the topic is, can we breed a better bee, improving New Zealand's honeybee stocks through science and industry cooperation. I'd like to welcome Peter Dearden from Otago University. Peter trained as an evolutionary developmental biologist, focusing on the evolution of traits in insects. He's worked on many species, but fascinated by bees. He's Professor of Biochemistry Department of the University of Otago, Director of Genomics, Aotearoa, a national network that aims to improve the use and value of genomics in New Zealand. And he also leads the Future Bees breeding program, which is the focus of the presentation. So welcome, Peter.
Yes. Hello. It's bad up here. The lights are shining in my eyes. So if I look really angry, it's just because I can't see any of you. You are still out there, right? Good. So my name's Peter Dearden. I'm really proud to be here. It's great to be invited to come and talk about this program. I guess I wanted to start by talking a little bit about the Future Bees program and what we're trying to achieve. You may have met some people involved with the Future Bees program or heard of us or even given us bees to measure genetic diversity. So if you've been involved or you've had some bees taken from you or something like that, you had a talk with Gertje, for example, who is going to stand up right now. So Gertje, who does most of our wandering around the country. Then thank you very much. We really need that industry engagement if we're ever going to succeed in doing what we're trying to achieve. So Future Bees is an MB-funded genetic improvement program. This program is running for about 5 years. We're about a year and a half in, so I'm not going to have solved all your problems yet. Gives a couple of years. Sorry, AFB seems like a tricky one. One of the things I wanted to point out is that this program started with actually a collaboration between the University of Otago, Abacus Bio and Taylor Pass Honey Company, so I would like to thank those guys, but the program we have is funded directly by MB. It doesn't actually have any beekeeper funding in it, though we have a number of partners who I'll talk about a little bit later who are contributing their expertise and their skills and hopefully access to some of their bees. So we are working with industry, but the point here is that we kind of felt at this end of the work we wanted to see what we could achieve before we started to ask you for money. So, I press a button that says next. Okay, so the aim of the... Nothing happened. That happened. Okay, so the aim of the Future Bees program is to develop and apply tools to integrate modern genomic selection into honeybee breeding, enabling New Zealand beekeepers to rapidly improve honeybee performance, right? So I wanted to point out that I'm a geneticist. I like to hang around in my lab and play with bees and other insects. I have absolutely no wish to run a honeybee improvement program for you guys. What we're trying to achieve here is to develop the tools and technologies so you can run your own honeybee development improvement program. Okay, so that's the point. We want to develop the tools that are affordable, effective, and useful such that you can do it yourselves. And we want to leave, after our funding runs out, the industry with that possibility. So that's what we want to do. I wanted to start by talking a little bit about why we think this is important and show you some examples of what actually selective breeding could do for you. So we know that in most production industries, in fact I can't think of a production industry where this isn't the case, that selective breeding brings gains in productivity and efficiency. Better stocks produce better outcomes. And a lot of the time when we talk about better outcomes, we're talking about more efficient outcomes. And I think looking at the state of the industry today, it looks to me like there are some hard times ahead. And so efficiency is critically important to you. I think this is not the time to say, well, actually, this is something that we can't afford. This is something which may be able to produce more with less input. We also know that modern technologies using genomics in particular, so studying the genetics of the entire genetics of an organism rather than a few genes, allows you to do better, faster, and more accurate selection. So it improves that speed and accuracy, enabling you to quickly make gains in your stocks, which would have a benefit out there in your operations. And that's been demonstrated in many other industries, and I want to show you an industry where this works. This is, I think, one of the most remarkable achievements of New Zealand science that nobody ever talks about because it's about sheep, and New Zealand talks too much about sheep. So I just wanted to show you this table, which is basically looking at, oh look, there's a little red line here. What we're looking at here is the change in performance of the New Zealand sheep industry. Much of this is due to a strong selective breeding program which started in about 1990, and using tools that were available then. Now the current New Zealand sheep breeding is using effectively whole genome techniques to select sheep. So in 1990, there were 39 million adult ewes mated. In 2015, there were 19 million. So there's a huge drop-off in the number of ewes that are out there. Despite this, we're looking at a 30-fold increase in the number of lambs produced, right? So we've got many less sheep on our hills, but they're producing as much or more lambs. So these numbers all go up. It's amazing. So live weight of ewes have gone up. Lamb carcass weight has gone up. Carcasses sold per breeding female is up almost 100%. Right? So these are remarkable increases in gains, and if we look at this fantastic graph here, I love to show graphs because they're usually inexplicable. Again, we're looking here at total number of sheep in green. The number of sheep have gone down. So it's gone down quite drastically, but we're actually selling about the same amount of total lamb. All right? And a lot of this is due to strong selective breeding. There is a huge component of working with those stocks to get the best out of them, but actually selective breeding to ensure that we're getting the right number of lambs per ewe and that we're making the best use of each of the ewes that we have has made an enormous difference. Okay? That's what we want to achieve for bee breeding. But bee breeding is difficult, right? So sheep are kind of easy. They sort of do stuff that we can manage really easily. Bees aren't, okay? So there is a lot of science involved in moving these sorts of selective breeding programs into bees. And I wanted to go through some of these things and talk about some of the solutions that we have generated or are generating to kind of get you there. So as you know, bees are not sheep. They are much cuter. I shouldn't make jokes. It just puts me off, right? So, and there are some real issues. So, in fact, most selective breeding programs involve inbreeding, right? So, when you look in your flock of sheep, you're picking the ones that you like the best. You're breeding them over generations. You're effectively inbreeding those sheep, but enriching for genetic variation that gives the traits that you like. Now, doing that in bees is a really, really bad idea, and you all really know why. And that's because of the fact that bees are haplodiploid, right? What we mean here is that they determine their sex on the basis of one single gene and genetic variation at that gene, and you all know this is the sex locus. As you inbreed bees, you end up losing your stock. So so if we have sorry, I'm looking at the wrong slide here. I'm supposed to be looking at that one. So if we have in our bee systems, in your beehive, there are a bunch of worker bees who are all female. You have a queen bee who's female, and you produce drones that are male. All of these guys over here are diploid. Just like you, they have 2 copies of each of their genome. All right? But these boys over this side, unlike you, have only 1 copy. And the way a bee tells whether it has 1 copy of its genome or 2 copies is genetic variation at one particular gene. And that one particular gene is called the complementary sex determinant gene. And if you get in the situation where you have a male bee which has the same copy of the gene as one of the copies in a queen, then the offspring, some of which will have the same 2 alleles, and those will turn into drones even if they have 2 copies of that gene. Okay? So that means if you inbreed bees, they have a special bonus effect for us geneticists that inbreeding them is terribly, terribly bad. Inbreeding sheep, you just get dumb sheep. Inbreed bees, they drop dead. Okay? So that's a bad idea. Our solution to this is actually to generate genetic tests where we can determine what 2 alleles a queen has and ensure that when we're doing crossbreeding schemes that we are maximizing the number of alleles in a population. That enables us to do genomic selection and do that inbreeding while maintaining diversity at this gene. Okay? So we have a bunch of tools to do that, and they sort of work, and they're almost affordable, which is nice. Oh, look, dead bees. Good. Sorry, I stole Gertrude's slides because I'm very, very lazy, but so sometimes they surprise me by doing animation-y things. The second thing is that bee biology is challenging. Right? One of the big challenges for us is actually understanding the parentage of a particular bee. So again, if we take a sheep example, if we have a population of sheep, it's really pretty easy to understand the pedigree of those sheep. We know for each sheep who their mother is and who their father is. So we can use genomic information and use that pedigree to do our selective breeding. We can also take a flock of sheep and say, if we look at the genetics of these sheep, we can understand how related they are. And these 2 kind of measurements are very important in selective breeding. Of course, in bees, we often can't do that, right? So we usually don't know who the parents of a bee is. We might know the parentage of a particular queen, but we normally don't know what their male parents are, because bees mate with multiple males on the wing. It's really hard to keep track of that. So this leaves us with this problem, that actually the only way for us to determine the relationships of bees is to look across the whole bee genome and say, if we sequence all of the bee genome, can we compare those sequences and use that to reconstruct those pedigree relationships? That's really quite tremendously difficult, and we've been trying to do that for some years, and we've switched to the fact where we've realized that actually the only way we're going to achieve that effectively is by sequencing every piece of DNA in each honeybee that we're interested in. So we've moved from sort of genotyping bees and thinking about a few genes to the fact that now, to look at a population of bees, we'll sequence the entire genome of a bee. If we want you guys to take up this sort of technology in your breeding programs, we need to do that cheaply. To sequence the genome of a human being at the moment costs about $2,000. We've got the cost of sequencing a bee down to about $40. So given that the first bee genome was sequenced in 2006 and costs over $3 million to do, I reckon we've done a pretty damn good job of reducing the cost there. That must be said, it wasn't really us. Okay, so next issue that we have to deal with is that bees are very sensitive to their environment. Sheep are pretty environmentally insensitive, but bees are very sensitive to the environment, and as a geneticist, this is a real problem. The problem is that, I'm going to put up a not very complicated equation up here, but the point is that if all of the variation you see in the performance of your bees is due to the environment, there's not much point mucking around with the genetics, right? Because we're not going to make the gains that we need, all right? So we need to actually work out which bits of the environmental influence. Okay, so maybe we'll explain this a little bit. It says here, phenotype equals genotype plus environment. So phenotype is what we say is the kind of traits of a bee. If you are looking at me now, as I'm sure you are, because I'm fascinating, this is my phenotype, right? It's disappointing, I know, but it is my phenotype. And you can think about particular traits that I have. Phenotypically, I think the technical term is fucked eyesight, right? That's one of my phenotypes. These are traits that we can look for in bees, not obviously those traits, but the traits that we can look for in bees, we call these phenotypes. These phenotypes, for example, my eyes, all my family need glasses, right? So there's probably some genetic component there. But also I spend a lot of time hunched over papers reading extremely complicated bits of work, and that's basically damaged my eyes. So there's a relationship with both my genetics and my environment. If the environment is a critical thing, messing around with the genetics is kind of pointless. So one of the things we're trying to do is work out how much of the variation we see in beekeeping operations is due to environmental things, and particularly environmental things we can change. So we're looking at ways of assessing nutrition in hives using simple tests and assessing pathogen load in hives. And it's good that we've been talking about qPCR tests for AFB. Those are the sorts of things that we're looking at. Can we put those things into our breeding programs to say, actually, the genetic gains here would be much better if we were managing those environmental influences? 18 minutes. Sorry, you can't see that. There's a clock here saying 18 minutes. It's making me nervous. The last thing that's problematic is that phenotyping bees is hard. So again, we want to measure the phenotype, the traits of our bees. And we need to do that in a way that we can consistently compare across operations that this bee or this hive is producing, the genetics of this hive is producing this kind of quality output, is producing this amount of honey, or etc. And that's a very, very difficult thing to do because of that environmental influence. Because of the challenge for beekeepers of actually going in there and recording information, I find it very difficult to fill in forms while wearing a bee suit. I know you guys do too. And so we're trying to find ways to automatically phenotype things that we're interested in. Okay? And we're trialing a whole load of things, including automatic phenotyping with various equipment. Not necessarily the ones on the slide here, but lots of things that we're trialing. Just seeing if we can actually get really decent measures so that it's not a burden to a beekeeper to really measure the traits that they need for these kind of breeding programs. Okay, so those are our difficulties and the ways that we're developing technologies to do that. So the first half of this project is really developing those technologies. We're doing the genomics. In fact, Josh sitting down there in the hat is sequencing honeybee genomes on a daily basis, I think. We're developing the CSD tests. We're developing the Goethe and others around the country are developing ways of accurately phenotyping bees with these kind of new technologies. But I wanted to put some things forward which I think need to be said because it's very easy to misconstrue what we're trying to achieve here. And in a few moments, I'm going to ask for your help. which is what we desperately need. So one of the things that I'm always asked about this program is, oh, you're going to find genes that provide resistance to, for example, AFB or varroamite, etc. We are not. Right? That's a really interesting scientific thing to do, and I would love to be funded to do that and discover these genes and become rich and famous, but that's not the point. Okay? We are trying to find ways to make an economically better bee, not one that has particular traits that you think you might be interested in. Okay? The reason for this is actually most traits that are important to you are not governed by a single gene. All right? Stop looking for the gene that provides resistance to varroamite. There isn't one. It will be polygenic. There will be multiple genes involved. And that means that once we've found what those multiple genes are, we probably can do bugger all about them. Okay? The same applies to, for example, the two twin diseases which are really causing havoc in our population, obesity and diabetes. Right? We have studied for years genes underlying obesity and diabetes. What do we find? There are thousands of genes involved. All right? Best bet is to skip McDonald's. Genetics isn't going to help you. Okay. The other problem is that when you start taking a particular trait and selecting just for that trait, you ignore all the other ones that are important. Okay? And genetics is a trade-off. Okay? We are not. This is not X-Men, right? We can't find a mutation which allows you to fly. If we did find such a mutation, it would have a trade-off, and those trade-offs are important. So all selection involves trade-offs. While we might be able to select bees which are more resistant to AFB, by concentrating on that one factor, we may make those bees unable to make as much honey, or we may make them more aggressive, or we may make them more susceptible to varroamite. None of which are outcomes that I think you'd particularly want. So it's important to us to know what trade-offs you are happy with. Okay? We need to be able to say, if you, in your operations, what is the characteristics of a bee that would be best for you, and then we can select for that. So instead of concentrating on the biology of are they resistant to this or are they able to do that, we have to concentrate on the economics in your operations. What is the value of a particular bee to you? And in fact, the selection matrix that we use is asking questions about economic value. It's not asking questions about genetic value. We want to get bees which are more valuable to you. That may mean that they produce more honey, but that may have a trade-off for other particular things. It fits with your operation and what you want to do. So in a moment, I'm going to put up a website which I want you to go to and tell us what sort of bees you would like. It's no good me deciding what sort of bees you would like. I would like bees that were friendly because I'm allergic to them, right? And don't sting. That's probably not beneficial to you. We want you to tell us what you need. Then we'll achieve it. And then we will try and achieve it. There we go. So, the steps of this project are pretty simple. And I may rock through this pretty quickly because I want to give you time for questions, and I know I'm sort of in the way of lunch. But the first thing that we're doing is defining the genetic merit of particular bees. All right? So when we're doing that, we're basing our definition of merit on their performance. All right? And their performance could be a load of traits that you are interested in. Again, we need your information as to what those traits might be. And the other thing is their genetic relatedness. Okay? So if we can identify performance and their genetics, then we can start saying what families of bees produce the outcomes that you particularly like. All right? So we're all good with those genetic relationships. We have developed a whole load of things which will allow us to tell us those genetic relationships and enable us to define the genetic variation in the honeybee genome, and then we can relate that to the traits of performance that you particularly want. Oops. We've done this on a large scale, so many of you have been involved in our genetic diversity surveys. So we have been looking at ways of genotyping queens. So obviously the most important individual in a hive in terms of the genetics is the queen. So actually we need to know what the queen's genome looks like, and we've been doing that by looking at the genotypes of the drones that they produce. We're getting to the point where we're thinking we have a technology where we can sequence one individual drone and maybe a pool of 8 drones, and that would give us all the information we need about the genetics of that particular queen. With that information, we can start building relationships. So this is a figure from Goethe, which I particularly like because it's vaguely colourful. It is also completely meaningless. It is effectively a family tree of bees. You might like it in that format, right? This is a family tree, that format. That's a family tree of lots and lots of bees. But we can start then putting production traits on that family tree and say which ones of these groupings of bees have particular traits. And those are the bees that we could go to a beekeeping operation saying, okay, so you do want something which is slightly more resistant to AFB. Well, this family has that kind of that kind of characteristics. That's the family you should breed from, etc., etc. We're all good with that. That's working well. We then need to take those bees and select and breed the best candidates. We work with an elite queen breeder, Better Breeze Research, so that's great. It's great to work with an established bee breeding program, which is already doing some selective breeding, though not using genetics at this point. So we're working closely with them, but the technologies we're developing are not just for them. Right? They're technologies that we will gift to the whole industry, not just to Better Bees. And we're working with genetic consultants Abacus Bio. And Abacus Bio are a huge part of this program, and they are extremely experienced in dealing with genetic selection in other production industries in the country. So it's delightful to work with them, and they're quite funny too. Working with them has demonstrated to me the importance, because they have long experience with developing breeding schemes for other production industries, both in New Zealand and overseas. And the first thing they said was, well, what we actually need is to develop a breeding objective. Okay, so a breeding objective is really the goal of a breeding program. What are we breeding towards? And I think this is a really important point and something that we need your engagement on. A breeding objective is a way for us to say, okay, what do you want and how are we going to achieve that? But by knowing what that endpoint is, knowing what we're breeding towards, that enables us to bring all these technologies to bear and perhaps produce something that you might actually want. So what we're trying to do is define the ideal individual that you want in your operations. Okay? Now, it may be that different operations want different ideal individuals, and we're happy with that kind of complexity. So if you're mainly doing pollination, it may be that you need different bees from those who are mainly doing honey production. But that's something that we're going to tease apart. Basically, when you think about a breeding objective, there's here's three random characteristics which we think are important to the New Zealand industry. So first one is honey yield. We should be selecting to increase honey yield from bees. That would probably be a good thing for many of you. Gentleness is something that many people are interested in. We have reasonable evidence to suggest that more gentle bees are actually more productive at honey, and they're certainly easier to work with. They reduce your time at an apiary, and they're much more fun. And winter survival, right? We're very clear that actually bees that die during winter are probably not economically efficient for your operations. That's where your costs are. So we can see that those are three key things that we would like to improve. If we could improve honey yield, gentleness, and winter survival, that would probably be a good thing. But these are just things that these come from Goetze's PhD, who has looked at the value of these particular things in an economic sense. But actually the question is, what do you want in a queen? Are you interested in reducing swarming? Is it increasing honey that you're particularly interested in? Winter survival, body color, varroa resistance. These are all traits that we could manage through genetics and genomics, but we need you to know about, that you need you to tell us which things you want. So if you go to our website, which is FutureBeesNZ, get your cameras out, take a photo, come on, wordpress.com. There is a link from that website which will lead you to a long and complicated survey. You may get annoyed with the survey. I do. The survey seems to ask you the same question endlessly, but the point is to ask clearly what things you are willing to trade off to get the bees that you think are economically valuable. Okay? So we can't just say, do you want improved winter survival? We want to say, do you want improved winter survival, and would you do that at the expense of varroa resistance or at the expense of honey production? Right? So by asking you to trade off those things mentally, we can get a better picture of what people want. The survey will also ask you what sort of operation you run, because it's very clear that different sorts of operations may want different sorts of bees. We hope to generate that complexity, and it's a big part of what we want to achieve. So we need you to engage with that, go on that survey. It's kind of fun. Take half an hour. How long does it take? Half an hour? 25 to 30 minutes. And many of you could race through it. No, get some coffee and some cake and put your feet up, get the computer out, spend 25, but think about it. Don't rush through it, think about it, because it's important to us to know what you want. So go to that website, or Gertrude has hundreds of cards which she can give you which have that website on it. So go and mob her afterwards, vigorously. Probably not in hashtag MeToo age. Okay, 6 minutes, awesome. Okay, the next part of the process, we're developing those genomic tools, we're developing an understanding of what a breeding objective is. The next step is to test the improved genetics that we generate in commercial operations, and we have a bunch of partners who we are going to be working with to test those things. So Taylor Pass Honey Company, thanks guys, you guys have been awesome. Tai Tokoro Honey, who I've only ever met through email, so if any of you are here today, it would be great to say hi and shake hands. Midlands Apiaries, our friends down south, and Ngāti Porou Mea, who are also involved in the project. So all of these people have offered to allow us to use some of their apiary space so that we can actually test these bees and run proper testing to say, actually, not only is the process we've developed working, but it actually works in a commercial operation. We're also thinking about new traits. Are there ways that we can assess new traits that we could select for? And we're working very closely with Plant and Food Research, who I know are here somewhere. Where are you guys? Is James in the hall? Well, anyway, I can't. There he is, over there. Hey, he's hiding. Particularly looking at pollination-related traits, because we think we could improve pollination, so we're really working closely with them. And with Denature Diagnosis, we're working on ways of actualizing some of this genomic work in the industry, which is kind of cool. All right. The point of all of this in my last 4 minutes is this. As I said, I don't want to run a bee breeding program for you. I've got more pointless things to do, and I enjoy them. The point here is to leave you with a structure that you can use to improve stocks in your industry, exactly as happens in the sheep industry, where while there is a body that looks after the data and the selection process, it's the industry which drives it. We want to leave you with an accessible and affordable way to determine the quality of your bees and to support your decision making. And that's what this is all about in the end, is support decision making about which hives you're going to split, which bees you're going to breed from, where you might buy a breeder queen from. It's all about supporting those decisions to improve the stock of New Zealand bees generally. We want to provide you with a system to help you access the bees you need to improve your stock, right? In the sheep industry, there is an internal market where you can kind of say, this is the sort of sheep characteristics that I want, and you'll get an answer saying, okay, this is the semen you might want to use for not the semen for sheep, this is the ram you might want to use to breed from your sheep. The same could be achieved for bees relatively easily, enabling you to fine-tune the stocks of bees that you use in your operation to reduce your costs and increase your output. And we want to, if all large operations are doing all of that, then what we should see generally is an improvement in the national bee stock, a bee stock that's more efficient and robust and allows you to do more with less. That's what we're trying to achieve. We've got must be 4.5 years, no, 4 years to achieve that. It won't be easy, but hopefully with your help we can. I'm going to finish by saying there's lots of people involved in this program. Perfect timing, eh? There's lots of people involved in this program, so feel free to email us at futurebeesnz@zoho.com. That's our website again. We have this beautiful logo. This is an MB funded program, so the Ministry of Business, Innovation and Employment. Sorry, it's not a good idea when you fumble with the sponsor's name. The Ministry of Business, Innovation and Employment are fantastic people. They've funded this program. They see the need and they see the benefit that could arise from it. As I said, this work is led out of the University of Otago, but with really huge input from Abacus Bio, our commercial partners, Better Bees, Taylor Pass Honey, Tai Tokoro Honey, Midlands, Ngāti Porou, Plant and Food, and Denature. And I reckon I've got some time for questions, so I'm going to stop here and invite you to shout at me. Yes, good. Do you want to go up to the thingy so everyone can hear your dulcet tones?
Do you have any data to show on bee diversity you were talking about?
Yeah. So we have we have sort of preliminary data on that. We're waiting for a pile of data to come through. So Gertrude travelled up and down the country this year mainly, that was to collect bee samples to look at diversity in New Zealand. So what we have done in the past is we've looked at diversity of those particular gene alleles which control sex determination. We have that data. That data's been available for a while on a website. And basically that data says that there is quite a lot of genetic diversity in New Zealand bees. But we're measuring a single gene, and that's not an ideal way to do this because there are regions of the genome which may be more constrained or less constrained. So the survey we've just done recently, and we're waiting for the data from, is across the entire genome. So we should have a much better idea of diversity. I think it's a really interesting question, though, right? How much diversity do we have? And can we actually see evidence of different imports or different use of bees in, Carniolan versus Italian? I think that's a really, really cool question.
Can I just add on to that, please?
You're not supposed to ask me questions.
I'm not asking anything. Hi. Just on that, Frank, because I've been asked a few times now if Future Bees will support importation or if we want importation, and part of this genetic diversity survey that Peter was just mentioning is actually that I'm going to be collecting some samples overseas as well, just to make sure that we actually understand where the New Zealand population sits in terms of comparison to overseas genetics, because we're very aware, and Peter has talked about the inbreeding effects, that we are an isolated population down here. So at this point in time, we can't really answer that question. We are probably going to be a lot smarter in about 6 months' time.
It seems unlikely.
Peter, what will it look like, the breeding program? I mean, will someone send in a sample of bees from a particular hive they might want to pull into pollination or for honey production in a certain area? What's it?
Yeah, so I think that's a really important question, and something that we haven't worked through at this point. The models that I think we're using are things like the sheep improvement program run by Beef and Lamb Genetics, where effectively breeders will they will send in samples to an agency who does the sequencing at a reasonable cost. And that's related to the characteristics of those particular individuals. And then you have an internal market where you can say, okay, these are the traits I want. What particular animal will provide me with those traits, and what's its cost, right? So that gives you an internal market where actually the best bees, or the best sheep in this case, are more highly valued, and that gives so that economic understanding comes into it. So we hope actually it provides a way for people to much better manage their stocks, but also, maybe they go, I really need to change, I'm getting into pollination, I need to buy in some pollination related bees and really change the nature of their particular stocks. But the point is not to, the point I think with the sheep breeding industry, the remarkable thing here is that these are individuals who are thinking about their own operations and improving their stock, and in doing so they've improved generally the stock in New Zealand. I think that's the approach you want to take. Bees are particularly annoying in that, when your queen flies to be mated, it's probably picking up drones from the neighbourhood. So, you can spend money on buying that perfect queen, but when you start producing daughter queens and they start to mate, who knows what they're mating with. But if we can get in a situation where actually lots of people are engaging with the program, the quality of the drones up there will improve as well. So so hopefully it'll end up being something where bee breeders will take the brunt of the cost because they want to show the value of their products to those people who are going to be using those in the production industry. Yes.
It just seemed from your talk that there is a sort of a conscious decision to not use or to move away from genetic engineering in this program, and I was just wondering why that was.
Oh, gosh. I am enthusiastic about genetic engineering because I think it can bring real value. In bees, the technology is effective, and in fact, Josh sitting there is going to go and learn how to do genetic modification of bees in Germany. However, the regulatory constraints in this country mean that whatever he does, it's not coming back here, and we're not going to be doing this outside of containment in New Zealand. So have no fear that we intend to release genetically modified bees. The reason we haven't done this in this particular program is that the key thing for us are outcomes for beekeepers. If there's a regulatory hurdle which really doesn't depend on beekeepers, it's probably insurmountable. I also think many people here will be selling organic honey, and genetic modification and organic really don't seem to go well together just at the moment. So I think we need to be aware of those technologies. We need to engage in them so that we are able to detect genetic modification of bees if it comes from overseas, that we are able to generate genetically modified bees if we need to with acute disease threats, but this isn't the solution for this particular industry. I do think that selective breeding with bees hasn't been done effectively anywhere, I think, in the world. Maybe in Europe there are some good programs, and so there is vast potential for us to do some good with this. If other things come along, let's have the tools and technologies ready, but we don't need to have them released now. Good question.
One last question. Peter, one of the, I suppose the standouts of sheep breeding was that they not only improve the genetics of the sheep, but they improve the genetics and the selection of the grass they feed on.
Yep.
So it's a combination of nutrition and genetics. So are we looking at this with the future bee out there?
I think that's a really good question. It's something that we haven't, that we're not going to address directly in this program. We have got a PhD student coming who's going to look at nutrition and whether we can find markers for nutrition so we can explain variation in bees that is related to their nutrition, not their genetics, right? So part of our job is to explain why you get such huge variance with bees and whether that's an environmental effect. So I can see that as a kind of building block working from the bees to say, actually, what nutrition might they be lacking in a particular environment? Do we have a simple test where we can say, actually, these bees are not getting the nutrition that they require? And then we have the opportunity to turn that into what would you do in the environment to improve that? Yeah. I think the Trees for Bees program, for example, is outstanding in that it provides the other end of this, which is actually what trees will produce. Pollen and nectar, which is of benefit to bees. I think we probably need to go beyond that. And I think the possibility of actually using selective breeding in places like the Manuka industry to selectively breed strains of Manuka that provide more nutrients to bees to support colonies while they're making honey would be a fantastic idea. You know, tree breeding is slow and painful, though. Bees are much better.
Yeah. Wouldn't mind having a talk with you just for a minute.
Sure. Awesome. Good, I can go. Thank you. Excellent. Cheers. Thank you.